Hello colleagues. The primary objective of this talk is to share some of our major findings for this project. A comprehensive report was submitted to HotGrow last year, so without wasting any time, I'll jump right into it. The project came about after several reports from growers about plum tree deaths in their orchards back in 2015. The cause of the tree deaths at the time was unclear because trees that were dead or dying were of different ages and cultivars, and the distribution of dying trees within the orchard was not uniform. Anecdotal evidence suggested a combination of factors could be responsible for what was observed. Among those were bacterial canker, drought, and ring nematodes. Ring nematodes had been suspect in an outbreak in Franschhoek in the previous year, but the observations were not documented. Therefore, our hypothesis was that there could potentially be a link between high ring nematode densities and bacterial canker outbreaks. A bacterial canker survey was conducted and soil samples collected from 12 major plum producing farms spanning 5 regions in the Western Cape, that is, Klapmuts, Wellington, Franschhoek, Robertsons, and Simondia. Trees were evaluated based on the scoring matrix shown on the slide, where 0 was healthy or without symptoms, and 3 being above 75% of loss of leaves and stem cankers observed, or that the tree was dead. Other characteristic symptoms of bacterial canker infection were also noted, such as oozing of gummosis from stem cankers and branch dieback. In the interest of saving time, I will skip this slide, but kept it in the presentation for those who may be interested in my soil sampling and ring nematode quantification processes. The observations made during the survey were analyzed statistically, whereby the probability of observing bacterial canker infection in an orchard was evaluated using logistic regression analyses. The ring nematode counts together with the observations were analyzed using a general linear model. I'll start by discussing the results from the logistic regression analysis. Here we wanted to model the probability of disease infection, whereby the occurrence of a symptomatic tree was as a result of time of season, drought stress, area of the farm, choice of rootstock, and high ring nematode densities. We first had to assess if there was any evidence to support that a combination of these factors was associated with disease infection. Simply put, does our model fit the data? The wild test was found to be significant, which means there's evidence to support that the combined factors are associated with disease infection. Also, our model could explain 57% of the variation in our data. Next, we determined the effect of each of the factors in influencing the likelihood of disease infection. There we found that all factors could significantly influence the possibility of disease infection, just at varying levels of significance. Drought stress was significant at the 5% level. The odds ratio estimates give us an indication of how much each factor, as considered in the study, is likely to influence the probability of a plum tree to have bacterial canker infection, should there be a unit increase or decrease in the observed data points of each factor, and when keeping all other factors constant. I have zoomed in on the effects with the highest odds ratio estimates for each factor. For example, a plum tree experiencing drought is twice as likely to have bacterial canker than one that does not experience drought. Following the same convention, looking at area and rootstock, a plum tree in Klapmuts or one grafted onto Sapo 778 has a higher chance of having bacterial canker infection than one in an orchard in Robertsons or grafted onto Mariana respectively. A unit change in ring nematode densities was shown to only increase the probability of bacterial canker infection on a plum tree by just 1%. Due to the ring nematode analysis including both the ring nematode counts and observations, we opted to analyze these data using a general linear model. Shown on this slide are the factors and data points included in the analysis, with the number of levels considered in each factor and the values that represent those levels. First, we assessed our model's fitness. We found that our model fits the data better than the sample mean of the ring nematode values across all the categories. Therefore, we rejected our null hypothesis for the alternative. This is shown by the significant alpha value. Also, our model could explain over 50% of the variation in our data. We then proceeded to determine the effect of each of the factors in explaining the differences in the ring nematode values. We found that all except drought stress were significant in explaining differences in the ring nematode densities. However, since our data was not balanced, and by that I mean that the data points in the analysis for each factor were not equal, pairwise comparisons of the least square means were conducted to explain where the differences in the ring nematode values occurred. Shown on this slide is a diffogram plot. The range on the axis is determined by the largest and smallest least square means. The dashed diagonal line is a reference and acts as the slope of the plot. There are vertical and horizontal reference line segments which are on the plot. These represent the pairwise comparisons. If the line segment, as shown on the slide, the blue line segment, intersects the dashed diagonal reference line, this indicates that the pairwise comparison of the least square means of the ring nematode values for the respective levels within each factor is not significant. And if the inverse is true, where the line segment, that blue line segment, does not intersect the dashed diagonal reference line, this means that the pairwise comparison between the least square mean is significant. On this slide, I have shown a diffogram plot of the pairwise comparison of the least square means for ring nematode values between drought and no drought stress. Because the blue line segment does not intersect the diagonal reference line, this indicates significant differences in the pairwise comparisons, whereas previously there were no significant differences between the ring nematode values of plum trees experiencing drought stress and those that did not. Also, there were significant differences from the pairwise comparisons of the least square means for ring nematode values between Robertson's and the 4 other areas surveyed in the Western Cape, as shown by the 4 blue line segments. None were observed in the pairwise comparisons of the least square means between the 4 other areas, as represented by the ring line segments, which are intersecting the diagonal reference line. Lastly, there were no significant differences observed in the pairwise comparisons of the least square means of the ring nematode values between disease-free and infected plum tree, as indicated by the red line segment intersecting the dashed diagonal reference line. Again, this supports the evidence that there is a weak to no association between the presence of high ring nematode densities and bacterial canker outbreaks. Just some concluding remarks. The factors considered in this study, though not extensive but important for initial consideration, were found to have varying significant impact on both the likelihood of bacterial canker infection and ring nematode infestation. Drought and area seem to have the most significant effect on ring nematode infestation and chances of bacterial canker infection. I suppose this speaks to the various climatic conditions that these plum trees are exposed to at any given moment, which may become conducive for disease development. As an initial consideration at trying to provide solutions to what was observed back in 2015, our study did not reveal any significant link between ring nematode infestation and bacterial canker infection, as had previously been reported. We would like to thank in particular hot grow and plum growers for making this study possible. Please feel free to contact my supervisor or myself should there be any questions regarding our findings or for possible future collaborations. Thank you.